Thanks for joining today for the Whole Farm Sustainable Pest Management course. This is the third lesson of 12. Our mission is to promote a healthy, viable agriculture that protects and restores wild nature, and we help bring nature back to the farm. Our 2050 vision is we would love to see 1 million nest boxes and perches on farms. Birds, avian pest control is part of the toolbox for managing crops. Another part of our vision is hedgerows to the moon and back, and more than just hedgerows, habitat that helps support all the different kinds of beneficials on the farm. And we also want to see riparian areas be restored because they are the lifeblood of the landscape. Next. Well, I'm Joanne Baumgartner, Executive Director of Wild Farm Alliance, and I wanted to point out we have a lot of different resources that you can check out related to pest management. I'm going to point these out today, our video resources. We create mainly really short videos, 5-minute videos where we interview growers like Phil Foster on the left with researchers. And with that one, it is researcher Tara Pisani-Garreau, who is actually going to be speaking in our 5th webinar. And he talks, or they talk a lot about hedgerows and the benefits. And then this image on the right is from Steve Mathiasen and Jill Klein's vineyard up in Napa. We have interviewed Steve and researcher Houston Wilson. Houston spoke at our first webinar. So today's webinar is Managing Pests with Bio-Pesticides. We have Matt Griship, who is the founding director of the Grimm Center for Organic Production and Research at Cal Poly, San Luis Obispo, and Rex Dufour with the National Center for Appropriate Technology. I want to just give a heads up for our lesson after this one is going to be about augmentative biocontrol modes of release and types of supportive habitat. And it'll be with Kim Horton of Taylor Farms and Hannah Call of CAF. Next. Well, please consider attending all the other lessons that are coming up, and they go through March. We have continuing educations from DPR and the American Society of Agronomy. So lesson 3, we are going to start with Matt Griship.
Thank you so much, Joanne. I really appreciate that. If anyone can't hear me, just please let me know. We did a sound check, but computers are goofy. So as Joanne mentioned, I'm the director of the Grimm Family Center for Organic Production and Research. We've been in operation for not quite 3 years at Cal Poly. Our mission is really to provide education and research to support California organic industry. So, oh, I'm sorry, I have to ask you to advance. I just tried to advance. Awesome. So I've split today's presentation to 3 parts. So I'm going to introduce a concept I've been playing around with called the insecticide triangle. And if you have comments on it at the end, I'd love to hear them. Then I'm going to talk about pesticide performance of some of our traditional chemistries. So the chemistries that we've been using since really World War II to the present day. Then I'm going to talk about bioinsecticide performance. So we'll contrast how different groups of biopesticides or bioinsecticides compare to performance characteristics of our traditional insecticides. All right, so as promised, we're going to take this fast because I don't want to talk too much. But this is this triangle for insecticide-based insect management that I've been thinking about for a while. And this is shamelessly stolen from the plant pathologist disease triangle that many of you may be familiar with. So the idea here is that if you're going down the route of using an insecticide as an important part of your pest management scheme, there are at least three sort of categories that I would encourage you to think about when you're making your applications. So the first of those is insect biology, and that would include everything from the insect's physiology to potential resistance, behaviors, life stages, phenology. Then there's the application method, and by that I'm really talking about how are you delivering the insecticide to whatever your target is, whether that's directly the insect or whether it's the plant you're trying to protect. And then the last, and the one I'm really going to spend time on, is the insecticide performance characteristics. So what do I mean by insecticide performance characteristics? Well, I've broken this into 5 categories. You could add or subtract them, I'm sure. The first is really spectrum of activity. And so there, what I'm referring to is how broad a range of different types of insects does the insecticide or bioinsecticide interact with. The next is the speed of action and potency. So this is how fast does it either kill or otherwise mitigate the pest, and what kind of a dose do you need? And I think this is important to think about. So pyrethroids, for example, a lot of them, when you look at labeled rates, you might only be applying 0.05 pounds per acre of active ingredient, which is kind of mind-blowing to me that such a small amount of material spread over such a large area could have such a large ecological impact. Next, there's delivery pathway. This is different than application method. This is really thinking about, okay, the product's out there, how is it actually getting into the insect's physiology to mitigate or kill or however it works? Then there's residual. This is really about time. How much time does it protect what you're trying to protect for? And then, of course, cost is always a factor. So is this a really affordable product, or is it more expensive? So we're just going to go through these thinking about sort of classical insecticides. So really here I'm talking about chlorinated hydrocarbons, organophosphates, carbamates, pyrethroids, neonicotinoids, which are more about 1990s and up, and those classes of insecticides that have really been sort of the dominant and most used insecticides in agriculture. So all of those are very much broad spectrum. They kill a wide range of insects. Pretty much all those compounds I listed will kill your target pests. They'll also kill your pollinators. They will also kill any natural enemies or biocontrol agents that you're trying to rely on. And a lot of that is because of how quickly they work, and that's really tied to, they're all nerve poisons. So these things attack a physiological system that is widely conserved across insects. Insects have pretty similar nervous systems across orders, and they work at pretty low doses also. You don't need a lot of molecules of these compounds to monkey wrench the insect's nervous system. The next category is delivery pathway, and I'm just going to talk about the top 3 here. These are 6 classic delivery pathways though. The first one is direct contact. This is where you actually spray the compound onto the pest. Not a very common insecticide delivery pathway. The next, which is most common of those compounds I talked about, is secondary contact or residual uptake. The idea here is that the insect, as it moves across the protected plant, is actually picking up the compound through its cuticle. And all those compounds I talked about are very lipophilic or hydrophobic, meaning they like fat. Insects have a wax layer on the outside of their cuticle, so they very much readily work their way into the insect's physiology. In terms of residual, a lot of the compounds I've talked about have traditionally given in many systems anywhere from 2 to 3 weeks of activity, which means that you're only applying them, you're not applying them very frequently. And this is important because if you think about trying to target phenological stages of insects, if something lasts a long time, it gives you a lot of sort of slop room. If you apply it a little too early, not a big deal. If you apply it too late, that's always a big deal. And then the majority of these compounds are petrochemical derived, and they're derived through high pressure chemistry, actually chemical processes that really saw their origin with the Haber-Bosch process and the Germans' synthetic gasoline processes that they used to turn coal into gas in the Second World War. I bring that up because these large industrial processes are very scalable. So it's very easy to create a lot of product cheaply once you've got that upfront investment in a large chemical factory. So that means that these tend to be pretty, the old stuff we used tended to be pretty inexpensive. So you're thinking, you know, $10, $20 an application. So now I want to go back to this insecticide triangle, and what I'm going to posit to you is that this combination of performance characteristics has really allowed us to really rely on those performance characteristics and not have to think quite so much about insect biology. They're very forgiving. Also, I've worked in spray application technology, and from a coverage standpoint, if something kills at a very low dose, it kills through contact, and it lasts a long time, spray coverage becomes less of an issue. So you don't need to think we haven't needed to think as much about application method as maybe we will. All right, so now I'm going to transition and we'll compare and contrast some common bioinsecticides. I also might use the term reduced risk and their performance characteristics. So there are there's different ways to describe bioinsecticides. A fairly current one is to break them into 4 different categories. We're really going to talk about microbial insecticides and biochemical insecticides. Plant incorporated protectants, that's basically a fancy way of saying GMO, so like BT genes in a plant. And then RNAi is a kind of a novel technology that's really just starting to work its way into the industry. For specialty crop ag, which is most of California, really microbial and biochemical are the 2 categories that you're most likely to interact with. Next slide, please. So I'm going to start by talking about just a few different categories of microbial pesticides. Something that I think is always really important to remember is these are living organisms. So you've got bacteria, fungi, nematodes, which actually aren't regulated like pesticides because they're animals. However, we use them like pesticides, so I tend to lump them in here as do a lot of entomologists. And then viruses. So these are all living organisms, which means they're fragile. So you really need to be careful and look at the label and look at storage. You know, if you have a lot of temperature shift, that may deactivate these organisms, or that's a fancy way of saying kill them. You also want to think about your tank mixes. For example, applying a fungi along with, say, a copper or a sulfur as a fungicide, probably not going to be a good mix. But needless to say, they're living. This also means that once they're out in the environment, you have to think about the environmental conditions you're putting them out in, because they're not going to be tolerant of every environmental condition out there. So I'm going to start and just talk about a few groups of these. The first group, and I'm just going to refer to active ingredients here, are entomopathogenic fungi. So these are fungi that attack insects. The most common one in bioinsecticides is Beauveria bassiana. This is a soil-living fungus. It likes moisture like most fungi. And the way it works is it produces conidial spores. This is what's harvested and grown in the lab and used in formulations. They're sprayed out onto a crop to protect it. When the spores contact the insect's cuticle, they actually go through a process that's really similar to a lot of plant pathogenic spores. They produce an aprosorium. They push a fungal sort of finger into the insect's body. They produce mycelium. They produce a toxin. They kill the insect. They then reproduce in the insect. And then the lower figure shows you all a bunch of unfortunate critters that have sporulation coming out of them. So what does this mean in terms of performance characteristics? Well, the first one is it broad spectrum? And this is a little tricky with Beauveria because different strains have different activity spectrums. Some are a little broader than others. The general concept for most products is that they're pretty selective. They mostly they do best on softer bodied insects and herbivores a bit better than most natural enemies and pollinators. The next performance characteristic is how fast do they work? They're slow. It's a disease. It has to infect the insect. So you're looking at day or days before the insect is actually going to succumb. And that might be important if you're trying to manage, say, a vector. They do provide secondary contact like those traditional insecticides. Basically, it's actually contacting the spores that leads to the kill step of the insecticide. They tend not to have a long residual. These are living organisms. They're a soil organism. When we put them up into the canopy, they get exposed to a lot of ultraviolet radiation. They don't like that. So again, you want to read your label, but a good rule of thumb is, maybe 7 days of residual control. And then finally, they're quite expensive. These are compared to our traditional insecticides. These have to be grown and packaged. It's not a cheap process. It's not nearly as scalable as the chemical processes used for traditional insecticides. So next we can talk about nematodes. Again, not regulated like an insecticide, but we use them as such. These are much like the plant parasitic nematodes that you may be managing in your crops. They are nearly microscopic roundworms in their juvenile infective stage. The way these work is in the top of that little cartoon, they actually hunt for an insect, usually in the soil. They enter through the breathing apparatus or spiracles on the insect, the mouth or the anus. They can also enter through wounds on insects. They release a bacterium once they get inside the insect, which sort of putrefies the insect, ferments it, and then they start to reproduce like crazy in it. And then infective juveniles burst out of the what we call a cadaver of the insect. There's a poor waxworm at the top of there, just showing you the number of nematodes that are coming out of that. Those are actually probably adults because they're pretty visible. In terms of performance characteristics, again, there are different species and strains within species, and their activity spectrum varies a bit. They tend to be less broadly active than the synthetic organic nerve poisons, but they can be fairly broad. They're really best applied to the soil. They like a moist environment, just like the fungi. Just like the fungi, they're not as quick as a nerve poison. They're going to take a day or two to knock the pest down and kill it. It's a biological process. They're actually better than secondary contact, I would say, because in a soil environment, they actually will seek out insects. So when you mass release these onto your soil, you're getting sort of like a hunter-seeker kind of smart insecticide, if you will. If you can maintain a moist condition and if you have the right temperature range for the strain you're applying, research has shown that you can get up to 2 to 3 weeks of residual nematodes in your system. Unfortunately, they're incredibly expensive. Organic formulations run upwards of $300 per acre at a 500 million to 1 billion nematode application rate. Conventional formulations are a little bit cheaper, but you're looking at potentially hundreds of dollars per acre to apply these. Their biggest success has really been in greenhouse production, where you have a much smaller mass of soil that you're trying to treat. Bacillus thuringiensis is a tried and true microbial biopesticide. We've been using this for over 100 years. It's also a soil organism, also very prone to UV degradation. This bacterium has to be ingested by the insect. It produces a crystalline protein, which if the pH is just right and the digestive system is just right, will basically kind of cause sepsis in the insect. It perforates the midgut, allowing bacteria into the main compartment of the insect, which leads to death. Very selective. Different strains attack different orders of insect. The most common strain in ag use is Kurstaki and Azawa. These attack lepidopteran or moth larvae. So this is a larval stomach poison. It works best if you can target young larvae. Older larvae tend to be more resistant to Bacillus thuringiensis. It is slow acting. Again, it's a disease organism. This must be ingested, so it's a stomach poison. So an insect just walking across this is not necessarily going to get a lethal dose. Again, as a living organism, expect maybe a week, depending on the amount of heat and light in your system. And unlike the other ones I've talked about, these are actually pretty cheap. They're fairly competitive with some of the, probably the more expensive synthetic chemicals. So now I'm going to talk about biochemical pesticides. I skipped over viruses because there's only a few examples of those. And this includes things that are produced by living organisms like plant extracts or microbial metabolites. It can also be pheromones, which are actually synthetic, but the origin is all biological. This sometimes also includes things like sulfur, copper, well, copper not so much for insects. Those sometimes get lumped into this category as well. So I'm going to talk about 2 plant extracts. The first is pyrethrum. Pyrethrum is a nerve poison. So would you advance, please? And the mechanism of action with pyrethrums is that, like DDT and many of the chlorinated hydrocarbons, it interacts with the sodium channel on nerves. So it actually monkeys with individual nerves. It causes overexcitation and eventual death. So it comes from chrysanthemum daisies, which are largely grown in Africa. There are records of people using chrysanthemum daisies in pest management that go back, I think, at least a millennia. Broad spectrum, contact active, just like a pyrethroid, provides secondary contact, just like a pyrethroid. Very short residual. So pyrethroids are synthetic and they have functional groups that have been modified so that they stand up to UV degradation. Not so with pyrethrum. If you're in a hot, warm environment, you can expect 12 to 24 hours of residual in many systems. Typically, that means you want to apply them in the evening to maximize coverage. And they tend to be expensive. Again, not as scalable a technology because you actually have to grow these massive crops of chrysanthemum daisies to get enough product to sell. So the next plant extract is neem seed oil, and the major active ingredient in neem seed oil, which is azadirachtin. So the neem tree is native to India. It's also it widely grows in Africa. It grows in Central America now as well. The insecticide is extracted from the covering of the need seed. It's an oil expressed from the seed and the outer pithy stuff around the seed. The way it works is actually as an insect growth regulator. So azadirachtin interacts with insects when they are molting between stages. So either larval stages or from larvae to pupae, pupae to adult. So what this means is that, interestingly enough, this works best on soft-bodied insects. There's a lot of research supporting that neem is fairly selective, especially towards pollinators and some natural enemies. It doesn't act fast. It's not a nerve poison. Really, the insect has to get to one of those development stages to die. So it can take up to weeks to kill an insect. It may kill it in a couple of days. There is some repellency from neem as well. It also has some fungicidal properties. It does provide some secondary contact. It's an oil, so it can be brought up through the insect exoskeleton. Like a lot of these other products, as a natural product, it doesn't have the residual that our synthetic organics do. So again, read your label, but rule of thumb would be maybe a week. It's a little bit cheaper than pyrethrum. So now we're going to move on to a bacterial naturalite. There are a variety of these out there. The most commonly used one are spinosins. This is derived from a bacterium that was collected in the Caribbean from a sugar plantation. It's a nerve poison. Advance, please. In this case, it acts on the nicotinic acetylcholine receptor. So this is a receptor that interacts with the neurotransmitter acetylcholine. So this is a chemical that basically it's not a direct transmission like a sodium potassium part of a dendrite. It actually works between nerves, so it allows signals to move between nerves. Acetylcholine, if it's not stripped, basically overexcites the nervous system, leading to death. This essentially causes that. It is broad spectrum. Works fast because it's nerve poison. It provides secondary residual contact while wet primarily. And this can happen either from when you first apply the compound, re-wetting from dew. We don't get as much of that in California as maybe out east. But we did some research in my old lab where we showed that it actually reactivates contact activity with dew. Once it's dried down, it really becomes more of a stomach poison. In terms of duration of activity, this compound, it kind of depends on the pest you're trying to manage. If it's one that you really need contact activity from, it's going to be shorter. If it's one that is going to consume a lot of plant material, like a caterpillar, it could be a little longer, but 7 to 14 days, depending on your pest. There are label restrictions on rotational partners with this product, though. It's one of the few insecticides that has that, so you got to pay attention to that. And unfortunately, it's pretty expensive. So then there are the soaps and oils, and I'm going to talk very briefly about insecticidal soaps, standard sort of horticultural oils, and then essential oils here. And this is a diverse group. All of them share some common mechanisms of action. So potassium fatty acids are what we are in most of our soaps. A vegetable oil or mineral oil is most of our horticultural oils. These work by contact, so they actually have to be sprayed on the pest typically. The first way that they kill insects is they interact with the waxy cuticle, that waxy layer on the cuticle. They actually strip it, and then if it's hot and dry, the insect will dry out. The next way that they can kill insects is by suffocation. So that it'll actually work its way into the breathing apparatus or the spiracles of insects, leading to suffocation. For soaps in particular, they can also cause some cell death if enough of it gets into the insect, but there's a lot of arguments on how prevalent that is as a toxicological mode. For those two, the soaps and the sort of traditional oils, they tend to be really cheap. They don't really have any residual because you have to actually hit the insect pest with it. They don't have secondary contact as a result, but they can act pretty quickly. Drying out and suffocating can kill things pretty quickly. Horticultural oils are sometimes used to suffocate insect eggs. That can be really important in some systems. And then those products are really cheap. I mean, they're sometimes even cheaper than synthetic organics. Essential oils are kind of different. Next slide, please. So essential oils at a high enough dose can have those first mechanisms of action, but they also have some potentially neurotoxic activity. So essential oils like oil of cloves, geraniol, cinnamon extract, there's garlic extract, a bunch of them. Some of them actually inhibit the acetylcholine esterase biosynthesis. So acetylcholine esterase is a compound that strips acetylcholine off that acetylcholine receptor that we talked about earlier. So that leads to overexcitation of the insect. So you can get some secondary contact out of essential oils. They don't last very long though. They're pretty volatile and they tend to be quite expensive. It's not very scalable to extract a lot of cinnamon oil or other compounds. And then the last group I'm going to talk about are pheromones. And I'm going to focus just on Lepidopteran mating disruption, although there is mating disruption for some scale insects and mealybugs now. The way this works is pheromones are really important for different insects at different stages of their lives. For moths, for example, many moths, the female produces a pheromone, this rare compound in the environment. It allows males to find her, and then they can mate and have babies and cause you trouble. If we can synthesize the pheromone and put it out in the environment at a high level, it essentially creates a lot of noise, if you will, like static on a TV. And the male can't find the female. If he can't find her, there's no mating, no next generation. So if you do use mating disruption, you always want to remember it's got to be applied as a preventative. If you miss the behavioral, like mating or whatever you're trying to eliminate, it's not going to help you. Also, they rarely provide control as a standalone. Often, the most successful mating disruption programs I've worked in have always had another biopesticide component, either an ovicide or a larvicide. And then the last thing is, at least for Lepidoptera, we've shown time and time again, the longer you use this technique, the better it works. The lower numbers of insects in your environment, the easier it is to keep the males from finding the females. So if you continue doing it season to season, you're probably going to see an increase in efficacy over time. I love mating disruption because it's species specific. It's not really fast or slow because it's purely preventative. It's keeping insects from reproducing. It doesn't really fit the secondary contact mode. It has a very long residual. Hanging dispensers can last a full season. It tends to be fairly expensive though. Finish up, and I apologize, I think I've gone over a little bit. So, effective use of biopesticides. The reality of biopesticides is that we probably can no longer just rely on insecticide performance characteristics like we've grown accustomed to in many ag systems. Which means that you're going to have to think more about insect biology. You're going to have to think more about your application method. You got to know your pest. What's its damage potential? What's its phenology? What are the susceptible life stages to the tactic that you're employing? What are the potential delivery routes? Is it a stomach poison? Is it a contact poison? You've got to know your biopesticide. Are they compatible with your current delivery systems and the way you have those set up? You very well may need to go to a higher volume spray to get better coverage. You're probably going to also want to spend a little more time calibrating your sprayer. If you're using an air assist sprayer, you really want to think about your air velocity because that has a lot to do with coverage in those systems. Also, if you're looking at biopesticides, I always think it's really important if someone's trying to sell you a product and they can't describe the mechanism of action really well, be leery because it's really hard to target a specific life stage of an insect if you don't understand how this thing is supposed to do what it's supposed to do, kill the insect. And then finally, thinking about IPM thresholds, these products, I told you about pyrethroids and 0.05 pounds of active ingredient per acre for some of them. Bacillus thuringiensis, a high rate is 2 pounds per acre. So you really want to think about, can I keep enough residual out there? Do I need to be more conservative in terms of when I begin my program? Because these things just typically don't provide the oomph that their synthetic counterparts do. And then finally, really thinking about your insect community, which is really what this whole thing is about. What pests can be naturally controlled in your system? Oftentimes things like aphids, leaf-feeding insects, unless the leaf is the saleable part of your plant. If you back off and can increase biocontrol, you can get some good economic control there. However, there's probably always going to be a few pests, so say like a codling moth, navel orangewort. It's really well adapted to your crop, and you're always going to have to think about managing it, and a bioinsecticide might be a good tool for that. And I'm going to stop talking now and hopefully answer some questions. Thank you all for listening.
Yeah, how about we're running a little behind. We do have some questions, but let's first run the poll. And this is for the audience to we picked a couple of questions with Matt's help on what did he cover, and we'd like you to answer them, and then Matt will go over them. So we'll take a minute or so. So please answer these questions. All right. How about if we've got about 80% participation, and how about if we end the poll and Matt, can you go over these and see what people were thinking and what the right answer is?
Yeah. So the first question asks, compared to traditional insecticides, biorational insecticides typically have a broader target range, which is also true. And the have shorter residual effect compared to traditional insecticides is the correct answer. Some do work through contact poisons, but many are stomach poisons as well, or are stomach poisons. And very few of our biopesticides have fast kill and knockdown. Really, the two that I talked about today, it would be pyrethrum and spinosins. Everything else typically takes days. For the second, which type of pests are BTs most effective against? And this one's a little bit of a trick question. The most correct answer is caterpillars. We don't have any BTs that, actually, I think there might be bioengineering one for true bugs, but delivery is going to be interesting. I don't know of any that kill ant larvae. Moths and butterflies is sort of correct, but it's the wrong life stage. Basically, by the time it's an adult, the BT is not going to really do any good for you. They're not eating leaves at that point.
Okay, well, thanks, Matt. How about if we take one of these questions, we'll go on to Rex's and then take the rest of the questions at the end of the session. And so are there current BTs for diamondback moth, which is a huge problem in brassicas?
Yeah, that's a great question. So we have two BTs that have activity against diamondback moth. Currently, the one that seems to have the most support is BT Azawa. It would be the substrain. An example, can I say an example?
No.
Okay, I won't give an example.
I'm
The other, maybe Rex can get that. The other sub strain of BT, and it's really the classical strain, is Kerstaki. The problem is that diamondback moth. One of the reasons it's such a royal pain is that it is an absolute genius at insecticide resistance. And BT, there has been resistance to both of these strains of BT multiple times independently in different parts of the world. So honestly, one of my big concerns, because I'm an organic focused person, and that's really one of our big go-tos, is that as if BTs become really heavily used, we're likely to see resistance. And it really doesn't take very long for diamondback moth to exhibit that. Hopefully that answers the question.
Thank you. Thanks so much, Matt. That was so informative and interesting, fascinating. I love it. And just for the audience to know, part of this presentation, or a lot of this webinar series is funded by the California Department of Pesticide Regulation. And while they don't necessarily agree with everything or any of this of what we said, they since they are supporting the effort, they didn't want to trade names to be listed because they might unfairly get support that, yeah. Anyway, so hey, let's go to our next speaker. We're really happy to see Rex, or have Rex Dufour joining us. Rex is with the National Center for Appropriate Technology. And Rex, are you going to share your slides?
Sure. Thank you. My name is Rex Dufour. I work for the National Center for Appropriate Technology, which is a 501c3 nonprofit. If you have any questions about anything I say here, there's my email. I'm going to be talking, just introducing folks to NCAT and ATRA. NCAT is a 501c3 National Center for Appropriate Technology. ATRA is one of our big projects. It's a National Sustainable Ag Information Service, so it can provide, and you can contact it either through that website that I've listed or a toll-free phone number, and you can ask any questions related to sustainable organic crops, livestock, marketing, soils. You know, it's a pretty interesting service, and it's a free service. Couple things I wanted to mention. Some of the biopesticides they're pH sensitive. So if you're mixing with a high alkaline water, they may not work as appropriately as you would want them to. So just a note there. And also, here's a couple cabbage loopers that have ingested some BT. This is on a farm that I was advising for. They had loopers and actually cabbage moth on their brassicas. But go in, and if you apply a product, especially these biorationals or biopesticides, go in and check and see if they've actually done the job. Because if they haven't, you want to either spray again or switch to a different product. But this BT actually worked very well, even on these. These are third and fourth instars, like getting near to pupation, and the BT was very effective on them. So just wanted to mention that. So I'm going to be talking about a tool that's on the ATRA website. It's the Biorationals tool, and this is the ATRA website homepage. You'll see different pictures flashing across the screen. And I'm using screenshots to introduce this Biorationals page because I didn't want to wait for the internet. Can be a little bit slow sometimes. So I'm going to be using screenshots on this. So the Biorationals database, this is pretty user-friendly, and we tried to make it farmer-friendly. You can, and what I mean by biorationals, it's a slightly different definition than Matt was using, but it's three groups of low risk pesticides, either plant derived like pyrethrum or neem, or biologically derived from or microbially derived, viruses, bacteria, fungi. And then there's another group that I call kind of new wave pesticides, but that could be anything from pheromones to particle film barriers. And I won't mention the trade name, but it's a white clay dust. You might know what I mean. Um, so, and this database, you can search by pest type. There's 5 different pest types. And then that will generate a drop-down menu with both scientific names and common names. And we went that route because a lot of scientific names and common names are misspelled. And if you type in a misspelled common name or scientific name, you're not going to get any results. But I would caution when all else fails, read the directions. And that's what that says up there. So pest type and then pest name. And you always want to submit, click on the submit button, because if you don't click on the submit button, you're not going to get any results. But also you can search under pesticide trade name and also under active ingredient or active bio-ingredient. So let me just give you a couple examples here. So this is select a pest type. So there's disease, insect, mollusk, vertebrate, and weed. I'm going to select insect here. So I select insect, and that will generate a drop-down menu. It's alphabetical, and it's a very long list of insect pests. But I selected aphid because that's a very common pest. And so clicked on that, and it generates there's 96 results here generated. There's several pages of results. So there's a lot of what I call biorational pesticides that are registered for use on insects. And let me see. And the ones that are registered for use on organic production, you can see here this OMRI listed, that's Organic Materials Review Institute. If it says yes, it's generally okay to use on organic crops. If it says no, don't use it on organic crops. Okay, I'm going to move that over again. And so if you click on the third one down, the active ingredient azadirachtin, you can access all the products that have azadirachtin as the active ingredient. And so there are 2 pages, it's 21. So we have 21 different products that have azadirachtin listed. They're different trade names, different concentrations, and there's you can go to different websites, manufacturer's website. If you click on the trade name, which I will not mention, but the third one or the second one down, the third one down here, we're back to the main page. Third one down, in this particular biopesticide or biorational, it gives you some information about the beneficial organism or the active ingredient. And it also provides a listing of the pests it's registered for use on. Okay. So it's a pretty handy dandy tool. And just another note, there's a little button up here. it's pest prevention information. And that's important, especially for organic growers, because if you can prevent a pest from reaching an economic threshold, that is all for the good. Because once it's past the economic threshold, then you have to use some of these products, and they can be quite expensive, as Matt, I won't mention any names, but they can be pretty pricey. So if you click on the pest prevention, it will provide you with some information about, in this case, aphid prevention. We've only done this for the top dozen and a half insect pests, for just it's difficult to supply prevention information for every last pest, including weeds. So back to the main menu, I just wanted to show you, like, okay, this time I selected weed and then a weed pest, and then I selected bindweed because that's a fairly common and noxious weed, hard to control. And then don't forget to click on the submit search button. And then, as opposed to aphid, there's only 3 results. And that is kind of indicative of there's not a lot of good biorational products for weed control. I've talked to a lot of organic growers, and a lot of them said, I wish there was an organic Roundup. And I go, yeah, like, get in line, But there's only 3 products, and as I mentioned before, you can go up here, pest prevention, click on that button, and it's gives you a little bit of information about how to prevent bindweed. Anyway, play around with this database. It's a pretty handy dandy tool, I think, if you're looking for kind of a lower risk bunch of pesticides. We don't have methylparathion or a lot of we don't list neonicotinoids on here either, because they're broad spectrum and they have impacts on, they're not low risk. I don't consider them low risk. So a few resources. There's ncat.org. This is if you want to go to the Biorationals website and bypass the homepage. It's just atra.ncat.org and Biorationals. There's a, let me see, I'm going to move this over. Pictorial Guide to Hedgerow Plants and Beneficial Insects. That's an ATRA publication. It's also included in the excellent publication that Joanne's husband, Sam, wrote for CAF. It's Hedgerows for California. And then the Organic IPM Field Guide is another kind of prevention-based resource. And I think that's it for me. So any questions on that?
Yeah, thanks, Rex. Let's take one question that's simple about how do you use this, and then we'll do the poll. And that question was, can you filter by labeled commodity?
Oh, no, actually, you cannot filter by labeled commodity. We considered that, but this is the most complex database on the ATRA website, and we run several different databases, and it's been around for quite a few years. And it's no, you just can't. You can search by trade name, you can search by active ingredient, and you can search by pest type as well as, pest common name or scientific name. And those are the limits of the database.
Okay.
Good question, though.
Yeah. Let's run the poll now, and we'll take 1 minute. Here are 2 questions. please answer them, and then Rex will go over the right answer.
So we have 84%.
Yeah, let's
You think that's,
that's enough. Let's end the
yeah,
poll and
that's enough.
yeah.
Okay. I'll go over these questions, which are not biorational pesticides. And everybody got glyphosate is not a biorational pesticide. It's a very broad spectrum and effective systemic herbicide. Um, Before using the Sustainable Pest and Weed Control database, what do you need to know? And most people got that right. Determine the ID of the pest organism. If you misidentify the pest organism, then you may be selecting a biorational or some control method that is not very effective on the organism. So most everybody got that one right as well. So
Yeah.
good job.
Yeah, great job. Thanks, Rex. We have time for a couple more minutes. We have some questions for Matt, a few more. Matt, is there research output on how effective entomopathogenic fungi is on fruit flies?
Um, there, there's a lot of scholarly papers on Bovaria bassiana, and there's some more on Metarhizium, on a wide range of pests. I know that, so I guess I need to know what kind of fruit flies. So like Drosophila or Tephrididae. So so that would be like an apple maggot or all these invasive fruit flies that we're getting in California now, or Drosophila suzuki'i, the spotted wing Drosophila. Um, I can confidently say that for the latter, for Drosophila suzuki'i, not going to work. Um, yeah, just won't work. In terms of the tephridids, I think it probably depend on the individual fly, but there are a lot of papers looking at susceptibilities of these insects. The challenge is really getting the insects to interact with the fungal spores. So, if the fruit fly is kind of in and out of your crop, it might be pretty difficult to get a killing load of conidial spores on the insect. I think there's been some work done on attracting kill formulations. I think I've read some Indian papers where they were looking at bait stations where basically you've got a high concentration of the spores. The flies come in to feed on yeast or something, and they hopefully get a lethal dose. For all those fruit flies, once the larvae is inside your fruit, as my kids like to say, you're cooked. Um,
Okay, there's a couple other questions that are kind of related. Is there a trade-off with nematodes also attacking natural predators? And is there an interaction between microbial pests, biopests, and biochemical biopests?
oh, okay, okay. So for the first part of that question with the nematodes, um, it's interesting. So the more context the insect you have has with the nematodes in terms of its development and evolution, the less likely they're going to be effective because they've co-evolved. We did some research in my previous lab looking at a staphylinid beetle larvae that's commonly used in greenhouses, and found that it didn't do the nematodes were not very virile against the natural enemy compared to the fungus gnats and thrips pupae that we were trying to control. But there's definitely a potential for that. Again, the nematodes, they're interesting because there are some behavioral differences between the different species. There's also, it's just sort of like a different infective range. So for example, Steinernema feltii is a good one for those fungus gnats and thrips, and for smaller larvae. Steinernema carpocapsae is one that's been looked at for a lot of caterpillar pests, and there's a Steinernema rio brave that I know has been used pretty effectively against black vine weevil. And then there's Heterorhabdis bacteriophora. I'm sorry for the Latin. And And those have their own sort of range of insects they attack. But for all of those, I'd say, really, looking into, okay, what pest is this individual species good at managing is a really important first step. In terms of I think the second part of that question was,
Before, sorry, Matt, before you go there, I know I can remind you what the question is. I just want to say for the people who can't stay any longer, I bet you want to hear what Matt's going to say, but in case you can't, you can go to the link that's posted here to give us feedback in general and to give us your licenses if you need DPR CCA credit and to take that 10 question quiz for DPR credits. Thanks, Matt. So the question again was, is there an interaction between microbial biopests and my biochemical biopests?
so I'm guessing what that's getting at is if the living biopesticides could have an interaction with the non-living biopesticides that we were talking about.
Yeah, must be. Mhm.
Um, and that's a good question. I would think, and Rex, I mean, if you have knowledge or opinions on this, I'd love to hear it. It's going to vary pretty much by case to case. Um, like the nematodes, I don't think pyrethrum is going to affect them. I don't believe spinosins will affect them. Um, the other products, BTs, oh, I'm sorry, I don't know how an oilless soap might, because it could potentially monkey with them. They're pretty unprotected, and the fatty acids might mess with their outer cuticle. But I think when you read the label for those, it should provide you with some idea of, what you can and cannot ink mix with. I wouldn't think if you made separate applications, I wouldn't be as worried about it.
Yeah.
But if you tank mixed them, that's when I would really want to think about that. And Rex made a great point about pH. I left that out, and that's super critical, both for the livings and the deads. One thing that is incredibly important with the nematodes, so those of you who apply as a soil drench, you want to take the filters out of your system. And there should be label guidelines that'll tell you like what the the minimum micron size filter is that they'll go through. But otherwise, they'll just get shredded and they'll plug up your filter. Uh,
Yeah.
yeah. Hopefully, I answered that question. Okay.
Okay, yeah, there was, I'm just going to ask you the last question in case there's people that still can stick around. And that is, are biopesticides better at preventing pest resistance compared to insecticides? You kind of touched on that earlier, but
It really depends on the mechanism of action. So for example, Rex mentioned a kaolin clay product that works by basically irritating insects. It's a pretty good egg laying compound for a number of insects. Basically, they just don't like to be around it. They don't like to walk on it. They don't like to stick their ovipositor antennae into it. I don't think there's probably going to be a really big resistance to that unless it was some kind of weird behavioral resistance. For the nerve poisons, absolutely. I mean, they work on the same biochemical pathways as their synthetic counterparts. So, for example, those spinosins work on the same pathway as neonicotinoids. So there's a big potential, and there's been documented cross resistance. A pyrethrum works on the same biochemical pathway as DDT and pyrethroids. And so, again, cross resistance is a big potential. BTs, it's kind of species to species. As I mentioned, diamondback moth has shown a real ability to get resistant to that. Other insects maybe not. I didn't talk
Matt,
about. Oh,
can
sorry. Go
I
ahead.
can I
Yeah.
interrupt here? I've heard about because of BT corn, that
Yeah.
there's developing resistance to BT uh, in non-corn um, crops. So
Well, yeah, you're exactly right. Because I mean, if you look at like the current lines of transgenics, like have like 6 different BT Cris toxin stacks to deal with Western corn rootworm. So there's definitely a lot of potential of that. Um, for the nematodes, probably not. very easy for resistance unless it's already there. For the fungi, again, I mean, I don't, unless there's a lot of evolutionary context, I wouldn't think. So if that insect is evolved really closely with that fungi, then there's probably a good potential for it. If it's kind of a novel relationship that you're pushing, so like a foliar insect, and now you're putting this soil organism up there, I'd be surprised if there's a lot of potential for it. We've never had a documented, as far as I know, there hasn't been a documented case of resistance to mating disruption. I think there've been some controversial suggestions of it, but I haven't ever seen really good evidence of it. There, it's because if you think about, if you're a pheromone system, the female has part of that system. She produces the chemistry. The male has to have the right receptors to smell it. So any mutation to give you resistance would have to alter both of those things simultaneously. And given enough time, I'm sure it would happen, but I don't know if it would even happen within a human timescale. They've been around a lot longer than us. Like, insects have a 400 million year history. We have about a 400,000 year history.
Wow.
12,000 years of ag. So I'm going off on a tangent, though. I'm going to stop.
Yeah, yeah. Hey, well, thank you so much, Matt, for sharing all your knowledge, and Rex for sharing your really cool database that's so useful when people want to look at this stuff up. There's a place they can go. We also had, we put this link back up just in case you didn't get to it, to send us some feedback. And there are also resources on our page for this webinar. If you didn't get a chance to check those out, you should, because Rex and Matt recommended them. And please come to the next webinar. It's again on November 5th, which is a Wednesday at 11 a.m. Thanks so much for attending.